And so I'm recording this for our online buddies at the same time. So today we're talking about large biological molecules. So at the risk of only talking about food, what do people have for breakfast? Donuts? Yeah, delicious. If you had to categorize donuts as like a food group, well, I mean, okay, besides like tasty, what would you describe it as? Carbohydrates? Yeah, okay, cool. Carbohydrates? Yeah, donuts, yum. Somebody else? So sausage, yeah. If you describe this as a food group, what do you put it under? So protein, yeah, okay, so we've got carbohydrates, we've got protein, perfect. What else? Most foods you probably have breakfast. I need people to keep their masks on. Oh, yeah, thanks. I realize I'm paranoid, but we got this like email of doom from the school being like, you will keep your class's masks on, or otherwise everybody gets the plague and it's all your fault. I'm like, okay. Yeah. Anyway, sorry. Where was I? All right. Plague. Anyways, sorry. It's kind of weird. I realize it's a weird time because, like, no matter what you do, you're like, am I being paranoid? Am I being reckless? And either way, you just get to feel creepy all the time. But yay for us. Years from now, it'll be a cool story. Hopefully. Hopefully. You know, when you're like 80, you'll be like, back in the plague years. It'll be kind of cool. But, yeah. Okay, so you've got carbohydrates, you've got protein, probably some versions of fat, right? Probably there's butter or, I don't know, something that had fat in it. But those are kind of your options for things to have for breakfast. Carbohydrates, right? It could be bread, it could be cereal, it could be sugar in your coffee, whatever it is, right? It could be protein, it could be milk, it could be eggs, it could be meat, right? It could be peanut butter, and then it could be fat, it could be peanut butter, that kind of thing. The molecules that make up biology, the really big molecules, they're called macromolecules, are those options that you're aware of. So they're carbohydrates, they're lipids, so fats, they're proteins, and the other one that doesn't really come up for breakfast, but that is important, is are nucleic acids. They're things like DNA or RNA. Those are your four options for big molecules to make life out of. Okay? Comfortable with these options? You'd recognize all four of them? And of course, there's other stuff going on too, right? Like, there's water, which isn't any of these things. But it's not a massive biological molecule. It's just a little molecule that does things. Macromolecules are built from polymers. Well, so macromolecules are polymers built from monomers, which is a fancy way of saying, and hi people, I realize this is like a weird way to do it, but hi computer. If I've got little beads here, right? This little bead would be my monomer, just one. Like, if someone's monogamous, they only have one partner. Mono means one, okay? Monomer, if you get lots of them together, it's a polymer. All right, so as you string them together, you've got poly, you have many. You have a polymer. Many of your little repeating units. Are we comfortable with this idea? So far, so good? Theoretically, I can keep stringing these together until I get bored, right? Okay. So if I have beads as my monomer unit, I can have slightly different versions of beads, right? I can string some yellow ones on. They still connect. They're still the same type of thing. And it makes my bead pattern different than it would be if it was just all red, right? So far, so good? Great. So a polymer is this nice this molecule that has repeating units, lots of similar building blocks. Of these, of the 4 big organic molecules that we have, 3 of them, so everything except lipids, are made of polymers. Okay? So lipids are their own special thing. We'll get to them on Wednesday. But carbohydrates, nucleic acids, and proteins, all polymers. All right? So that means they all have a little individual chunk that's going to be repeated again and again. That individual chunk for each of them is a specific type of thing. But they can be slightly, the versions of the chunks can be slightly different. So, that analogy. For beads, they can come in different colors if you're stringing beads together in a pattern, right? If you're stringing food together in a pattern, right? These nice little kebabs. You're expecting food on each chunk. You don't want a bead somewhere in the middle of that. But the type of food you have on the stick can be slightly different. Yeah? If you're making little paper dolls, you're expecting paper for each of them, but you can have different colors of paper. Everyone's cool with this? No fair switching types of things between them. So no fair putting paper dolls into your kebabs, no fair putting beads into your paper dolls, none of that. Alright? If you're making a polymer of, if you're making carbohydrates, so a polymer of, we'll get to it, sugar, you can do that, sugar, long repeated pattern, but you can't start putting amino acids in there. That doesn't work. Okay? We're good? I'm going to harp on this. And this YouTube link that worked earlier doesn't, so I had to find a new one, but it's not as cool. But that's okay. And I close it down because I make poor choices sometimes. This is not super exciting, but we're just gonna, this is the one the other group saw, but this is not super exciting. Online people, I'm gonna find a better link and put it in your slides.
Types of polymerization.
Just want to look at the no, there are pictures? Nope. Fine. We'll look at the boring one. Don't memorize anything, just see the picture, okay?
In this lesson, you will learn about the polymerization of ethene. You will learn how to draw polyethene and the uses of this polymer. Polyethene is formed by the polymerization of ethene. Ethene is an alkene with 2 carbon atoms joined by a double covalent bond. Polymerization occurs when this double bond breaks at a temperature of around 200 degrees C and a very high pressure of 2,000 atmospheres. When the double bond breaks, adjacent molecules link together to form very long molecules of this repeating unit. The repeating unit was known as the monomer. So for example, in polyethene, the monomer is ethene. As you can imagine, using different monomers creates different polymers, with different repeat units along the chain, and therefore different properties.
We're comfortable with this idea? You can string this thing together, you can string this thing together, and they're going to behave differently. Alright? Cool. Great. Now, the basic pattern for how you string these together is fairly similar across different polymers. There are some slight variations, but the basic picture is this. So, you start with a little short polymer, or you start with just a single monomer unit, right? And note how this side has this OH group, and this side has this hydrogen here. Cool with that? This one has an OH group, this one has a hydrogen. Cool. Well, if a reaction happens and you pull this hydrogen off from the polymer, and this OH off, and you stuff them together, that's water, right? two H's and an O. You get off. It's called a dehydration reaction because you're losing water. Goodbye, water. And now these are stuck together. So far, so good. This slightly longer polymer now has an OH group over here, just like it did before, and has a hydrogen hanging around out here, right? Could you repeat this and add another monomer to it? Yeah, just over and over again. And you start pulling off water. You repeat this process where it's this dehydration reaction where these get linked together. These are now covalent bonds. It's a molecule, and it's longer than it was. You might have actually done this. Did anybody in school? It was really popular a few years ago. I don't know why, but it's cool. Mix glue with like borax. Did I do this? Yeah. What did it make? Like just properties wise. Squishy, hard, pokey. Squishy. Yeah. Okay. So if you've not done this, what you can do, it's fun. Should I have you guys do this? I don't. You mix borax in with water, and then you pour glue in on it, and as you stir, you end up making this like almost silly putty-like stuff. Yeah, slime. Slime's closer. You were the one who did it, right, Noel? Yeah, it's kind of slimy. What you're doing is you've got the little monomer of glue, right? And in the right conditions, if you add borax, it'll you can react it together. You can get them stuck together. These long chains, and now it's tougher. It's more like you can pick it up. It's not sticky quite so much. It's more slime-like. If you do this, if you do it in your dorm rooms or whatever, you'll notice as you do this, water comes off as you're mixing this. Water comes off. It's a dehydration reaction. You're losing water as you do this. Okay. If you do the reverse, you break this stuff apart. So this reverse would be, you have this polymer, and you add water to it, and you attach the hydrogen to this side, you attach the OH to this side, and you snapped off a chunk of this, right? So as I connect these two together, right, as I put two together, I lose water. If I put water into it and attach, I'll get that later. Attach hydrogen and oxygen to the ends, I'm breaking it apart. It's just the reverse of these things. For terms, just because sometimes there are a lot of terms in biology, it helps to know some of the roots. We already have poly, right, meaning many. Anybody comfortable with what hydro means? Water, right? Yeah. So like hydration, hydroponics, hydroelectric, right? Water. This list part, when you see that, you should think about things being cut or broken. So hydrolysis, water cutting. You'll see this in like when we get to something called lysosomes. They cut parts of the cell up. Glycolysis involves cutting. We'll get to that. So far, we're good? Lost anybody yet? Lost your neighbor? Does your neighbor look confused? Because that's fair too. If like you're getting it, you're like, okay, the person next to me is just drawing question marks over and over and over in their notebook. Stop me for them. All right. Polymers, the concept is fairly simple. To snap them together, snap them together, snap them together. But you can have so many different options of this. You have a very small set of monomers, but you can have a huge variety of polymers, right? So if we're using the example of English as the example here, you have 26 letters to choose from, right? And nobody really cares about X that often anyway. So you've got 26 letters to choose from. But you can make thousands and thousands of words, right? Just the order that you put them together in. You can have a really small set of monomers, but a huge variety of polymers. We're comfortable with that concept? All right. Nucleic acids are a polymer. So that means they're this repeating unit, and they've got a monomer unit that just shows up again and again and again. That monomer is called a nucleotide. The nucleotide looks like this. We're spending the last third of this course on DNA and stuff, so we're just going to kind of briefly go over this. You got a nucleotide, that's its monomer. You string a bunch of them together, and you have a polynucleotide, which is another term for a nucleic acid. Nucleic acids use nucleotides as their monomer unit. People with siblings, do you have the exact same DNA as your little sister? Nope, it's different. Do slime molds have the same DNA as pine trees? No, they're different, right? We're okay with this? It's very different. No matter what type of organism you are, you're going to have different DNA from each other. But the individual chunks that you get to make the DNA out of are going to be the same. So for DNA, you have 4 different kinds of nucleotides you can choose from. You've got T, A, G, and C. Those are your 4 choices. Those are your only choices for DNA. All right? So huge variation between individuals, but you only get 4 monomers to choose from. Right? The order they're strung together in makes a huge difference, but you still only have 4 individual building blocks you can stuff together. If you're still on the beads as the idea, you basically have 4 different colors of beads. Okay? And it's the pattern that's going to make the big difference. For RNA, it's also nucleic acid, also built of nucleotides. You have 4 options as well. The thing is, you've got the G, the C, and the A, the same as for DNA, but instead of T, you have U. It's a slightly different version of this, but again, only 4 options for RNA. Okay? So if somebody were to ask you ever, hanging around at a party, and someone's like, hey, what's the monomer unit of DNA? I realize this is not how parties work, but if somebody asks you, what's the monomer unit of DNA, what do you tell them? This is a party person, what are you talking about? But what would you answer if you needed to answer the question? This thing, right? This nucleotide, that's its individual chunk. Okay? We're good? Great. So that's nucleic acid. Hugely important. We'll see it a bunch. Not now. We are now going to talk about carbohydrates. Carbohydrates are amazing. Tell me about carbs. We eat them, we like them, they're cool, right? Okay. Carbohydrates are going to do a couple things. In biology, they're typically going to be fuel and building material, both. All right? So carbohydrates is a catch-all term that means sugar and polymers of sugar. So sugar can be a little monomer unit, and you can also make a big polymer of this. All right? So the most straightforward version are just one little sugar, just hanging out, a single unit, right? They're called simple sugars because they're not that complicated, but you can string them together into polysaccharides, which makes it way more interesting. So this unit here is called glucose. You've probably had glucose before, hopefully. I'll be willing to bet you've had glucose before. It's this simple sugar right here. That ring thing, you've got some groups coming off of it, right? If you get simple sugar glucose, and you get another simple sugar glucose, and you pull this oxygen and these 2 hydrogens off, and you link them together, that's a dehydration reaction. That's now formed a polymer. It's currently just disaccharides, just 2 of them. And they're stuck together. This linkage is called, this connection is called glycosidic linkage. They're basically holding hands now. Okay? All the same molecule. This is maltose. All right? If you do it, but instead of having the exact same sugar twice, you have glucose and fructose. You stick them together. that is sucrose. That's the kind of table sugar that you eat typically. It's two simple sugars stuck together with a glycosidic linkage. All right. You could imagine right here. Do you see this little OH group in this hydrogen here? And over on this side, same deal, and they're linked here. The same pattern's happening here. So you could put another sugar here, right? You could just keep doing this forever and ever and ever. You could just snap them together. When this happens, well, it's a polymerization, but when this happens, you end up with a polysaccharide. The type of polysaccharide you have is going to depend on what the individual chunks are. Are you using glucose as the are you using this glucose as the main building block? Are you using fructose? Are you using something else? And also how it's branched. So starch is glucose over and over and over and over and over and over and over again, right? So you've got this long molecule that's got lots of glucose chunks. And then if you want glucose, you snap a little portion of glucose off because you have this long starch molecule. If it's straight, it's called amylose. If it's slightly branching, it's called amylopectin. But again, it's just glucose over and over and over again. In your muscles, you store glucose. Not tons, but enough. You store it, though, as something called glycogen. It's glucose over and over and over again, but now it's a little bit branched. Concerns about this? Is it okay? All right. I want to look at this slide first. So, up there we've got starch. Glucose, 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 again and again and again, right? Down here, this is called cellulose, this stuff. This little thing, this is glucose, right? Believe me, this one's glucose, okay? Is this the same thing? Yeah, it's the same thing, right? Glucose. But instead of up there in starch, they're all facing the same way. And this one, they're flip-flopped, each other one. Do we see that? Okay. Starch and cellulose are both made of the same stuff, right? Glucose. But how they're put together is slightly different, and that makes very different properties. Does anybody, you don't have to tell me this if this tells you too much about, this probably tells you more about my childhood than you want to know, but have you ever tried to eat paper? Okay, so like, I remember reading some book about some spy or whomever who has like a secret map or secret letter or something, and they're about to get caught, so they chew it up and swallow it. Okay? You've heard this idea before? If you try to do that, it does not go well. You're there to be like, rah, rah. It is really hard to eat paper. Don't try it. You probably figured this out. Don't try it. Okay? As was apparently not obvious to me, but obvious for the rest of you, cellulose, the stuff that trees are made of, is tougher than starch. All right? The main reason it's tougher than like it builds trees or something is this pattern here. So cellulose, it's a polysaccharide, right? So lots of glucose over and over and over again. But the glycosidic linkages are different. So here we've got them flip-flopped, right? And this hydrogen can hydrogen bond really pretty well if it had something, an electronegative atom to hang out with, like an oxygen. So what you end up doing is you form hydrogen bonds. You've got this long chain here, and there's a hydrogen here that feels the electronegative atoms here, and you have them in sheets, and you connect them that way. Does that make sense? Now it's a lot tougher because the molecule's holding on to itself, but now they're also holding on to the other ones next to them. Does that concept make sense? So it makes it way more structurally sound. You can build redwood trees out of this stuff. But that's not the only reason you can't eat them well. The other reason is, the other reason is the enzymes that break down starch are looking for a specific shape to cut. Alright, so they're enzymes that can cut starch in portions. It's looking for a specific shape. And the ones you have look for starch. They're like, yep, cut. Okay? You don't have enzymes that can recognize cellulose. They come up to it and they're like, it doesn't fit. The way I'm trying to cut this, I can't get my scissors around that link. Alright? Can you think of animals that can digest cellulose? So they could eat grass and be fine? Cows, right? Yeah, horses are like, yeah, I could eat lots, right? Some animals can eat the cellulose. To digest cellulose, you need to have the enzymes that can make this cut, and this cut looks different than that cut. Concerns? Questions? Great. Don't go yet. We still have more of class. I had originally, in the slides, I said that we're going to do lipids today too. But that's a whole other topic. We're going to stay on carbohydrates. We're going to stay on polymers right now. Okay? So lipids, yes, are an important biological molecule. They're not a polymer. We're not going to talk about them today. Polymers are nucleic acids, carbohydrates, and then proteins. Okay? Yeah. And the single most common mistake I've seen with this, and I get why people do it, but the single most common one is people mix up what makes what. Okay? And I realize, as you're learning it, they all basically sound the same. Wow. Is that thunder? Wow, okay. Yeah, people are like, oh, it's going to be one of those days. You guys are fine. It'll be good. If the Wi-Fi goes out, though, that's going to be a problem. Just a sec. So this is all we have officially for lecture. I'm going to keep explaining more stuff, but online people, that's where we're at.